Okay, hello everyone. Welcome to Concordia University's Forest Space. Thank you for joining us for the next event in the SynBio Expressions series. This event series is Democratizing Science Through Conversations, and today's event, SynBio Conversations. To help situate you, we are streaming to YouTube live from Forest Space, and we are located on unceded Indigenous lands here in Tiohtià:ke, Montreal. And here at Forest Space, we work with the university community to help mobilize knowledge by co-creating daily activities. We're running today's event as a live stream Zoom meeting, so we welcome any comments or questions there via the chat. And those of you here in the space, just raise your hand, we'll get a microphone over to you, and the interviewers, as Bertie was just walking you through, just take care to speak into the microphone for us. Okay, with that, it's my pleasure to hand it over to our co-organizers for this series, Kathleen and Zoe. Welcome back.
Thank you. We'd like to open by acknowledging where we're sitting. We gather today in Tiohtià:ke, the ancestral and unceded lands under the custody of the Kanien'kehá:ka Nation. When we discuss the institution of science, it is especially important to consider the difficult history of science and indigenous knowledge systems and how it has shaped science methods. Science has, for a long time, attempted to distance itself from the context around it as to avoid bias, and in doing so has neglected to address anti-indigenous sentiments that have historically affected its funding, its findings, and its use. More recently, initiatives featuring scientific collaborations with autochthonous groups have resulted in scientific projects that address real-world needs and incorporate indigenous and community knowledge. The work is long. We encourage you to reflect on how colonial roots have shaped science and the future avenues ahead. A commitment to democratization cannot be complete without a commitment to decolonization. While science and technology can bring exciting developments in health, food, and the understanding of our natural and physical world, these systems can also be used for extreme destruction. Science, imperialism, and militarism have been entangled at the technological level, but also at an ideological level which seeks to establish science as an apolitical activity. Democratizing science entails acknowledging the politics of science and for scientists to step into a peacemaking role. We invite our audience to think of the ways science and technology have played a role in the current crises around the world, including the Democratic Republic of Congo, Ukraine, and Palestine. We urge Concordia to completely divest and cut ties with weapons manufacturers such as Lockheed Martin. Previously, Concordia successfully divested from fossil fuels, and this precedent can lay a successful path to future avenues. We hope you keep these ideas close to you as we explore the different facets of SynBio democratization and that you carry them with you into your work beyond this space.
This symposium could not have happened without the support of the Graduate Student Association, the Center of Synthetic Biology, the SynBio Apps Training Program. As well, Orly Weinberg has been an invaluable mentor who has been there from the conceptualization of this project to the execution, and we can't thank her enough. We'd also like to extend thanks to the folks at Fourth Space, Bertie, Doug, Jack, and Isa, who have been amazing at helping us brainstorm what a good science event can look like, and who are responsible for all the technical details that act like the phosphate backbone to our event's DNA, not to mention the beautiful space we're sitting in right now. And finally, we would like to give thanks to Quebec Public Interest Research Group, the Biology Graduate Student Association, the iGEM team, Christina Zanza, Dan Gershkin, Anya Chernikov, and others who have shaped the very delicate porous membrane of our events and have helped bring it to life. Oh, it's so much, okay. So SynBio Conversations will feature 4 fantastic scientists: Emmanuel Senbunya-Kato, Love Shrivastava, Natalia Kermesi, and Jacqueline Beaumont, who are all leading groundbreaking research projects across a breadth of disciplines. We have the support of 2 up-and-coming journalists, Haley Corlin and India Das Brown, who are here to facilitate these interviews. And this event will highlight the importance of offering avenues for knowledge transfer to scientists in their early careers, as to develop the important skill set for sharing research with a non-expert audience. After the interviews, we will move into breakout discussions to imagine the futures of each field and reconvene once more to share our final thoughts. So for our first interview, we have Emma Cato interviewed by Haley Caroline. Yep, sorry.
Are we good? Cool. Hi, everyone. Thanks for joining us today. I'm Haley. I'm a journalism student here at Concordia, and I'm excited to introduce to you today Emma Sabunia Cato joining us from Paris today. Emma is the founder of SynBio for All. It's a student initiative to increase access to SynBio education for students across Africa. And hi, Emma. I hope you're doing well. Thanks for staying up a bit later to talk to us.
Hello, Haley. Am I clear?
Yes. Yeah, I see you. So the first thing I wanted to ask you is, could you just give us an overview of what SynBio for All does? What is your organization about?
Okay. Thank you so much, Haley. I hope I am still clear. My name is Emma Sevunya Kato. I am originally from Uganda and currently I'm in France for my postgrad study. I would like to start by telling you about SynBio for All Africa. SynBio for All Africa is an initiative to make synthetic biology accessible to all African students. Our mission is basically on synthetic biology to improve access of synthetic biology in Africa and the whole world actually, and to ensure that synthetic biology literacy can be improved. What we do basically, we do teaching of synthetic biology. We teach synthetic biology, and also what we do, we organize monthly webinars whereby we invite professionals in synthetic biology to come in and give talks about synthetic biology to the students. Basically, in summary, that is what SynBio for All Africa is all about. We are a free organization. Our services are free of charge. We don't require any amount of money. Yeah.
Thanks. Yeah. And I'm also wondering if you could give us a bit of an overview of what students who take your course do, what type of activities they participate in, because I believe that almost 800 students have taken part in your courses. So could you tell us a bit about what they learn and what they do in the courses?
Okay. Our students, what basically they do, what we do during our courses, the students enroll. After enrolling, we start by teaching them, like we had the beginner's course, then the intermediate course. We teach them. After teaching them, we organize for them projects, and they do projects. At the end, they present these projects to a team of instructors, and the team of instructors award marks to these projects. Also, we go ahead and organize some small competitions among small groups of students. We organize students like in groups of 5, and they compete amongst themselves in projects relating with synthetic biology. During these competitions, a student is supposed to describe the problem is trying to solve using synthetic biology, is supposed to describe the biosafety and biosecurity measures which are being taken on his or her project, and is supposed to describe the entrepreneurship part of his project, if at all he wants to change it into an entrepreneurial project. So it's supposed to describe for us what are the entrepreneurship parts during these small competitions which we organize. But in future, we hope to organize very big competitions like for the whole of Africa in synthetic biology. Yeah.
Yeah, that's great. I also wanted to just hear a bit from you about what inspired you to found this organization, and why you felt like it was needed.
Okay, the inspiration of founding this initiative, it started in 2022 when I was at the IGEM Jamboree in France. When we were at the IGEM Jamboree that year in 2022, Africa was represented by 5 teams from Africa. There were 2 teams from Egypt, 2 teams from Ghana, and 1 team from Uganda. During the awarding ceremony at IGEM, there is an awarding ceremony. Africa didn't get an award, but to me, I felt like the projects, more especially the projects from Egypt, were worth to get an award. So it hurt me so much. I decided to make a follow-up with the IGM organizers and the judges to inquire and know what was the reason as to why African teams didn't get any award. In the response, I was told that the knowledge which African teams were applying in their project was not enough to win an award. That thing had to hurt me so much. And I, as an individual, I started thinking of a solution which I can bring to Africa to ensure that the African students also have the knowledge which is on the same level with the students from other continents, such as Europe, such as Asia, such as North America. And through that, I got an inspiration and I said, no, let me start up the SynBio for All Africa initiative. I offer services for free. I call upon different people to join me and we do this. So at SynBio for All Africa, after getting this inspiration, I didn't make my dream a reality alone. I called upon different people from Africa and I made a team of African people, which is 15 people. And then also I started looking for collaboration from other continents. I got collaboration in the USA from the EBRC, the PhD students under EBRC in the USA. They joined me. And then also I got a collaboration from Europe at the DTU, Denmark Technical University, whereby I got some lecturers who joined me and they have been supporting in teaching the students. Yeah.
Yeah, that's really impressive. My last question, out of all the things that you've done through making the courses, searching out collaborations, getting to work with the number of students you have, is there one thing that you're most proud of that SynBio for All has done?
Yeah, I'm very proud that by the time SynBio for All was starting, we started with the beginners course, and in this beginners course we had only 50 registered students, and at the end we gave out 30 certificates for students who finished the beginners course. But I felt that SynBio for All has brought an impact because when we advertised the intermediate course, we managed to get 800 students registering, and they all did the course and finished it, and we have just processed for them the certificate. So what makes me proud is that SynBio for All Africa is creating an impact in Africa because currently, at least we have an increased number of students in Africa who know about synthetic biology, which was different initially before SynBio for All Africa had started. And we need to push this spirit not only in Africa, but to collaborate with fellows from all over the world, including fellows from Canada. We can collaborate with them and maybe we share knowledge on how the impact which SynBio for All Africa has done. Some dedicated people can also take maybe some advice from us and we also take advice from them. And they also work in another area of the world. But I'm very proud that SynBio for All Africa currently has improved lives of many students to an extent that we have managed to get some support for students. For example, this year, we managed to send 2 students at the SynBio Beta conference in the USA. And this conference, we had a discussion with John Campbell, who is the CEO of SynBio Beta, and he gave us 2 sponsored positions. And as SynBio for All Africa, we sent 2 students to attend that conference. And also in July, we managed to get a scholarship from Cold Spring Harbor Laboratory in the USA. And we called for applications from all over Africa because we were given one position which was sponsored. We called for applications from all over Africa, and one of the students has been attending that course in the USA. It's called the Cold Spring Harbor Laboratory of Synthetic Biology in the USA. So those are successful stories, make me proud of SynBio for All Africa. And I believe I and the team which I work with, we shall keep pushing hard to ensure that we improve the synthetic biology access in Africa. Yeah.
Thank you so much for joining us. I really appreciate it, Emma.
Okay, thank you so much, Haley.
Thank you. Bye.
Yeah, bye.
Thanks for that. Thank you for joining us, Emma and Haley. So next we have Love Srivastava, interviewed by Zoe Katz.
So Love Srivastava is a postdoctoral researcher in the Ehrlicher Bioengineering Research Group in McGill. His biomedical research focuses on investigating the different relationships between cells and their nucleus and their role on the onset and development of different diseases, including cancer and Hutchinson-Gilford progeria syndrome. We met as collaborators, and I thought that his research work was really, really interesting. So thank you so much for joining us today.
Thank you for having me.
I wanted to ask you first, can you tell us a little bit about how you first became interested in biomedical research?
Sure. I think that's a question I get asked a lot. So my father, he is a practicing doctor in India. So since growing up, I've seen him practicing medicine, treating patients, things like that. But on the other hand, there are certain diseases which are still uncured. Cancer is on top of the list, then comes HIV, so on and so forth. So, that ignited the idea of the biomedical research, or in other words, the bioengineering, if I can help the community in finding some sort of treatment for the diseases which are still uncured. So the idea started with cancer, and why specifically cancer? Because someone I loved in my family died of cancer, so obviously that obviously ignited the idea, and then directed my future towards cancer research. Or in other words, it was mechanobiology, of which cancer is an example, which we will talk later.
Definitely. Yeah, I find that a lot of scientists in biomedical research in particular have these kinds of stories where something happens, families or friends, and then we're driven to really take up this research. You were talking about cell and nuclear mechanics. What exactly is that?
Okay, so I'll try not to go too technical. So we all know our body is made up of cells, right? And cells have different organelles, of which nucleus is like the most important one because it houses the DNA. And DNA is the main genetic makeup of our whole body. If there is a mutation or something goes wrong with the DNA, then that eventually is reflected in the physiology of the cell, eventually the physiology of the body. So what happens in cancer is there is a phase when cancer cells starts migrating from one localized area to different localized areas. And for that, they need to squeeze out of the capillaries. But the nucleus, being the largest and the stiffest organelle in the cell, restricts this migration. So what I found was from literature, this is not my work, from literature I found that this migration could be possible if the nuclei become soft. So then comes, okay, if I'm talking about the nuclei becoming stiffer or soft, there has to be something which is regulating this, this stiffness or the mechanical properties of the nucleus. Then I came across a protein which is known as lamin, and I found that this lamin protein is underexpressed in like so many different types of cancers, which makes the nucleus much more pliable and easy to squeeze out of the nuclear pores. So now the question was, okay, cancer cells, we know cells are immortal, they never die. The nucleus is soft. is there a disease in which there is accelerated aging, which is completely reverse of cancer. In cancer cells, the cells are immortal. And then I came across a disease which is known as progeria, as you mentioned, where the cells, they accelerate. It's like an accelerated aging. The patients having this disease are mostly children, and they die with an average age of around 14, 15, where they show symptoms of a 90, 95-year-old person. So now, interestingly, what I found was that this disease is basically due to a mutation in the same protein, lamin. So that made me excited. Okay, lamin, underexpression of lamin is making the nucleus soft. And if there is a mutation in this protein, what happens to the nucleus stiffness? And what I found was that the nucleus stiffness in case of progeria was like drastically increased. So for me, the nuclear stiffness was like the center point of so many diseases, because nucleus stiffness, the nucleus is always in a state of tension or deformation inside the cell. And we can imagine, like, for example, we have a ball made up of steel or a balloon. If you compress with the same force, the degree of deformation will be different in both the cases. And we shouldn't forget that all the DNA is stored in the nucleus. So obviously the degree of deformation changes how the cell behaves. So that's what made me interested in this idea. And eventually we did find that the nucleus stiffness has a role to play in how many times a cell can divide, because that's basically is known as the cell proliferation.
Okay, so what I'm hearing is that when you have a cell, you also have the nucleus, and the nucleus is kind of a barrier for things that can come in and out, and depending on how hard the nucleus is, that means that the barrier will be more strong or less strong in allowing things like this deformed protein that can lead to things like immortality or accelerated aging. So what was a turning point in your research?
Oh, so the thing was, first, as I mentioned, we needed to identify a protein which actually affects the nucleus stiffness. Yeah, from literature, we do get an idea that lamin AC is a protein who is kind of responsible for the nucleus stiffness, but they were mostly completely like depleted cells with lamin or overexpressed cells with lamin. You don't have like a linear scale between the nucleus stiffness and the lamin AC. So that's, I would say, what we did was quantify the nucleus stiffness as a function of lamin AC. So my first paper, it's like the plot I have is, I would say, pretty significant in the scientific community, which relates lamin AC expression with nucleus stiffness, which eventually could be a reason for so many diseases, like progeria being one. But there are like so many diseases, for example, muscular dystrophy and cardiodystrophy. We all see that there is a mutation of lamin, this protein. So I would say identification of lamin as a contributor to nucleus stiffness, which eventually will regulate the degree of nuclear deformation was the turning point.
Okay, so this lamin protein that you found was able to allow more or less nuclear stiffness, which means a higher or a lesser degree of this like barrier to other things coming in or out. That's very interesting. But you were talking about how this type of research could be applied and create, alternatives and therapeutics in biomedical context. How do you see that happening?
So that's a good question, because whatever I'm talking about eventually nails down to nuclear deformation. But now if I'm saying nuclear deformation in body, you just cannot compress each and every cell. So what I believe, again, coming back to the question you asked, what was the turning point? It's the lamin expression. Because in biomedical research, it's relatively easy. I wouldn't say it's easy. It's relatively easy to change the expression of the proteins. So now by changing the expression of lamin, we can control nuclear deformation. And why that's important? Because some studies have indicated that the degree of nuclear deformation regulates so many factors going inside the nucleus, of which YAP, which stands for yes-associated protein, is one. So when YAP is present inside the nucleus, It's basically a transcription factor responsible for transcription of like 100 genes. So if we can manipulate the nucleus stiffness by changing the expression of lamin, then we can regulate the translocation of so many factors, of which YAP is one. So I would say right now gene therapy, which includes or which specifically targets changing the expression of lamin, is probably the one. Or in future, if we could somehow manage to deform the nuclei, one of which is using osmotic pressure. So what we do in our research, just to quantify the stiffness of the nucleus, we expose the cells to external osmotic pressure using polyethylene glycol. So what happens is the nucleus gets compressed, and we can imagine a stiffer nucleus deforming less compared to a softer one, and we just calculate the nucleus stiffness from that. But that's not possible in a human body, right? You just cannot ask someone to drink 1 glass of PEG daily in the morning. That's not feasible. So, but changing the expression of the lamin, because the nuclear deformation is basically controlled by 2 factors. Number one is the stiffness of the nucleus, and the second being the forces acting on the nucleus. If we cannot regulate the forces on the nucleus, obviously the second option is to change or alter or manipulate the stiffness of the nucleus by changing the laminaceous expression. So I would say right now, the most feasible approach for my research in terms of practicality will be to test if we can change the expression of the genes, especially lamin.
That's really interesting. And I mean, we've all heard about these experimental gene therapy approaches and how they're super promising in cancer therapy. So it's really encouraging to hear that your findings can be applied to that therapeutic platform and produce real change. My last question is more in terms of like sharing science. What do you think? Why do you think it is important to share your findings with scientists and non-scientists alike?
Sure. With scientists, it's an easy one because even whatever I mentioned were mostly from the literature review produced by previous scientists. Because even I get asked, what are your discoveries? Did you make a drug or something like that? Science is not always making a drug. Like, for example, Newton, he discovered gravity, but he did not make rockets. But if we would have not known what gravity is, it wouldn't have been possible to make rockets, right? So I would say I focus, or our lab's focus is finding basic research insights, which eventually would be helpful for someone with a clinical research background to make it more, much more useful for the scientific community. So, yep, that's why it's important for the scientific community. For the non-scientific community is to track them towards science, number one. And number two, I think there should be some social awareness. Like we all know what happened during COVID. And I'm guessing 95% of the people now know what a PCR is. But before COVID, nobody knew that. So I think knowing the basics of science is always advantageous because we all know what gravity is, right? You don't need to be a scientist. So what my target is that at least everybody should know what a nucleus is because that eventually helps you maintain a decent health because there are so many factors affecting the health of the nucleus. So yeah, focusing on the factors which can keep you healthy is equally important and would be significant for the non-scientific community as well.
Amazing. Well, thank you so much for meeting with us today. That was a really interesting talk. Thank you.
Thank you for having me.
Thank you for that interview. Next up, we have Natalia Kurmasi, interviewed by India Das.
Thank you, Catherine. So I'm India. I'm a student here at Concordia, and I'm here with Natalia Kermesi. She's a grad student at She Lab at Concordia, and there she is working with microfluidics and synthetic biology. Thank you for being here, Natalia.
Thanks for having me.
So could you tell us a little bit in very simple terms, just some stuff about your research?
So Right now, I'm a grad student in the SHE lab, and my research focuses on synthetic biology and microfluidics. So right now, the main part of my project is studying the cells used for lab-grown meat. So I thought maybe I could kind of explain what lab-grown meat is, because I don't know if everyone is exactly familiar with how the whole process works, but so basically lab-grown meat is when you take a small muscle cell sample from a cow, so like a little sesame seed size muscle cell sample, and those cells are then grown to a large number, so we can have a bunch of these cells. And then when they grow to a specific number, they start to actually grow together as a muscle cell. And when this happens, they're moved to a big bioreactor, which is basically like a big tank that feeds a bunch of nutrients and media to the cells. And in this bioreactor, they're growing in a three-dimensional way. Now, this is really important because this is how you get different kinds of lab-grown meat. So whether that's like a steak or something where you would have different layers of muscle cells, or let's say like ground meat. And yeah, so that's kind of how the whole process works. And the issue in the lab-grown meat field, or a big area of research, is trying to decrease the cost, because it's really expensive to do this whole process, especially because it's a new technology, so it still needs to be developed. And the main thing that's driving up the cost is the media, so kind of the food that we need to give the cells in order to grow. And so that's where my research is focused on. We're trying to study the muscle cells, and it's known that they produce certain nutrients like naturally. And these nutrients are the nutrients that are supplemented into the media, and that's exactly why the media is so expensive. And so what we're trying to do is we're trying to study the cells and see, can we get the cells to make more of these nutrients themselves, so we don't have to add them into the media to kind of decrease the whole cost. And so, yeah, so I'm kind of using microfluidics as a tool to help me study these cells on a single cell level. So looking at the cells individually, and then, yeah, that's basically what my project is about.
So what inspired you to take up that research regarding lab-grown meat?
So for me, I was really driven by lab-grown meat being an alternative solution to animal agriculture. I think there's a lot of ethics. There's a lot of problems with the ethical aspects of animal agriculture, as well as sustainability. And obviously, there's 7 billion people, even more, on this planet. And so finding a sustainable food system and something that's more ethical, I think, is definitely a big driver behind why I wanted to do this. I just wanted to try and contribute something positive with my research. Yeah.
Cool. And I understand that you also work with microfluidics at the lab. So can you tell us a bit about what that is and how it contributes?
So microfluidics is exactly what it sounds like. You're basically able to control very small volumes of liquid. And what we do is we have these little microfluidic devices, and you can kind of design different, the way that you design the devices kind of allows you to control the very small volumes of liquids in different ways. And so the way I use microfluidics is droplet microfluidics. And so we have these channels, and when you flow the liquids through, you can create separate droplets. And so what I do is I use this to be able to study my cells at the single cell level. So each droplet, when you have different volumes, different concentrations of cells, you can kind of make it so that there's one cell per droplet. So it's almost like one little experiment is happening in each droplet. And because it's very small volumes of liquids, you can make thousands of different droplets per second. And so you can go through a lot of different samples, a lot of different conditions. And so that's kind of how I use it as a tool, is to study the cells.
That's really interesting. And what other synthetic biology tools do you use in your research?
So synthetic biology, I guess you could say it's altering a cell's natural biology, right? And so a SynBio tool that I use is gene editing. So that means you can add DNA to a cell or in a cell's genome. So you can give DNA, you can take DNA away, and that really controls how the cell behaves. So for example, I'm using gene editing to increase the expression of certain nutrients in the cell and seeing how it reacts, or taking certain genes away. Can that possibly make the cell grow less? Can it make the cell grow more? And so really we're using gene editing as a way to study these cells and seeing if it's possible to make them behave in certain ways. And yeah.
And how do you think that these technologies that you're using in the lab translate into health or energy solutions in the real world?
So how I think they relate to, for example, energy solutions in the world. So like I mentioned before, the lab-grown meat is really, it's an area of research trying to find an alternative solution to animal agriculture. And so in terms of animal agriculture, I think it's definitely a very energy intensive system, right? You grow these animals, a lot of food, a lot of energy goes into that. And so, for example, the let's say 70% of all crops grown go towards feeding agricultural animals. But let's say the food that comes out of that is not making up for the 70% of food we're putting into it. And so I feel like that definitely is a big issue in terms of sustainability, as well as greenhouse gas emissions. I know the recent UN climate change report reported that animal agriculture is over 15% of global greenhouse gas emissions, which is definitely a big chunk. And so lab-grown meat kind of aims to, finding an alternative solution, because like I said, there's a lot of people to feed, and this is not the most sustainable way. And so lab-grown meat is predicted to produce 80% less greenhouse gas emissions. So I think I guess my project is a little part in that whole research field, but I think there's a lot of different areas of research that are trying to move towards this overall goal of trying to create a more sustainable, maybe more ethical world.
It's really interesting to hear about how many applications like what you're doing has in the world. Do you think that there's any main challenges in your research that you have, and how do you address those?
Probably the main challenges is that a lot of times when you do experiments, they don't work. And like a lot of times I think that you have to do an experiment like 10 times before it actually works, because a lot of times we're doing something that not a lot of people know about, or that like I can't just go ask someone, hey, like I don't know how to fix this, like how do I make this work? So I think a big challenge is I guess the not knowing. So I feel like with each experiment, I try and approach it with, well, can I learn from this? Or sometimes, my experiments will take like 5 days. And so like there's something different I do on each day. And at the end, if it doesn't work, I don't know, did something go wrong on day 1? Because biology is unpredictable. Or did something go wrong on day 2? So kind of, going through each experiment and trying to learn, I kind of think it's successful if I learn something from it. So I think that's definitely a challenge, is being very like attentive and trying to like break down what the issue is. Yeah.
Yeah, I can imagine that a lot of the scientists probably understand exactly what you're saying. Mm-hmm. So I know we kind of touched on like what your research is doing right now to help with the health and energy fields. Do you think that there's any exciting possibilities in the future that could contribute to that?
So do you mean with like lab-grown meat, or just like in general?
So just synthetic biology in general.
Well, yeah, I think like what really drew me to synthetic biology is being able to use it as a tool to help solve different kinds of problems that we face in the world. So even just, for example, again, using synthetic biology to create alternatives to fossil fuel drilling. So there's a lot of research also going into biofuels, so producing fossil fuels with bacteria or microalgae and so on, or as well as gene editing being applied to different kinds of cancer therapies. Like, you know, I think that is something that's really interesting because it can save a lot of lives. And yeah, so I think, for example, you have a patient who, let's say, has some kind of leukemia, and their immune cells, which are supposed to recognize cancer as a pathogen, are not able to because cancer is our own cells, right? And so what scientists have been able to do is you take someone's blood and you isolate their immune cells, and then you give the cells DNA to allow them to recognize the cancer, and then they can be put back into the patient, and then now the patient's able to fight off cancer. So I think that when that happened a few years ago, that kind of blew my mind. So I think that's definitely a really cool area of synthetic biology.
Okay, well, thank you so much, Natalia.
No problem, thanks for having me.
Of course.
Super insightful interview. Okay, last but not least, we have Jacqueline Beaumont interviewed by Zoe Katz.
So I am really excited about this interview. Jacqueline is a transdisciplinary biomedia designer, researcher, and artist. I don't know if you define yourself as a scientist, but I definitely think that title applies here. Her work looks at the relationship between biotechnology, sex, nature, culture, and corporeal materialism. She combines fiber arts, audiovisual media, and immersive installations with scientific research and biotech. She is currently an independent affiliate at the Milio Institute and a collaborating researcher at the Biointerface Lab at Department of Material Engineering at McGill University. Thanks for joining us.
Thank you for having me.
So I'd love to go on a little journey. Would you take us back to the beginning and tell us a little bit about the first time that you connected the dots between the worlds of art and science?
I think my first point of connection was, for me, I was always, I was a very lonely kid. I grew up always constantly ostracized, and so I found myself drawn to nature as it didn't have any bias associated to it, and I could just get closer to it. So I think as I got closer to biology, I understood more about myself and the world around me, but it also made it very apparently clear that there's only so many avenues for people with a specific trajectory of thought to come into science. And so when I first was trying to experiment into getting more activated into synthetic biology, I became aware of the much bigger structures that are underneath all of it. And so I think when I was given an opportunity to be able to explore the potential to create with science, influenced by it, and like having it come from a more situated corporeal place inside my own body, it definitely changed how I understood the ways in which synthetic biology, or these very small, tiny little chemical mutations that we make in life, can truly alter an entire world. Particularly for me, I think my own transition was a huge portion of it, where I physically understood an unraveling and retangling of myself in the world, and was more aware of how these sort of structures and symptoms express themselves in the world, which is biology. So yeah, I think that's kind of where my introduction into it came from. I did my undergrad here at Concordia, and I was sort of taken up into the Milieu Institute with Dr. Alice Jari and White Feather Hunter, and both of those truly iconic women have given me an opportunity to fully express myself and experiment with these sorts of systems and thinking. So yeah, I think that's where it kind of all started.
Yeah, following up on that, I know that your work looks a lot at different binaries, including, this myth of like the left brain and the right brain. How do you address those interactions in your work?
Yeah, I think the idea of dualism or dichotomy and division in nature is something that I've always had. Personally, I was born in one body and I was finding myself in another. So I had understood nature to be much more non-binary in its essence. And so when I was when I was doing advanced placement biology in high school, and I was learning about like very cold, hard divisions between things, it made me sort of question them in a more nuanced way as to how those things could appear like that, but underneath it, structure something much more beautiful than any form of dualism could provide. Yeah, I think that's kind of where it all started.
Oh, fantastic. I love the way in which you bring these sort of concepts that we deal with in everyday life, and you make a point of showing how really this is a biological matter, right? It's a conversation that we're having at a cellular level, not just as two different people sharing these ideas. So maybe you could share some specific examples that you worked in where these conversations have happened?
Yeah, for sure. I think probably the tipping point for me was when I started working on a project called post-binary genetic sequences. This is a series of tapestries that basically visualize and reorient the, in some cases, full genomic sequencing, and in some cases, whole mitochondrial sequencing of a variety of different organisms. At first, they were just organisms that I had a sentimental attachment to, and I wanted to understand them more as a body of data and reconfigure these sort of binaristic divisions of even just a codon of like A, C, G, and T, when minimized to ones and zeros in an informatic or bioinformatic context, there's so much nuance that's lost. And so for me, I wanted to kind of take that data and allow somebody to see the gray space in between that and all of the nuance that can come out of the things that don't necessarily fall into those binaries. So yeah, so it's a series of tapestries. Some of them are in CMYK and some of them are in black and white pixelations. And this theme in my work has really kind of unraveled into different installations and different types of experimentations on cloth, printing, laser cutting, engraving. And so all of this has kind of brought me to a new place in my practice, but I guess we can talk about that more eventually. But yeah, so this is kind of where it all started for me.
Amazing. I would love to jump to where it is now and ask you a little bit about your ongoing project, Wet Dreams, and where that came, how did that idea come to materialize?
Yeah, I will just quickly zoom on through all my slides. There's so much. Here we go. Sorry, I'm a very visual person. So Wet Dreams is a project that I was very graciously supported by the Canadian Council of the Arts to begin producing, I guess it was last year. It's an immersive audiovisual exhibition that basically utilizes a portable genetic sequencing device designed by Oxford Nanopore And through working with them a little bit more closely, we've been able to, well, I was able to finally connect the DNA sequencer to Touch Designer, which is an audio-visual generation software. And the project, Wet Dreams, essentially reconfigures the microbial environment in a hydromicrobiome and creates a metagenomic environmental sample of a dance floor. Particularly the air after many, many hours of dancing is extremely humid, and it's a space that has been, or a material that has been conjured by hours of dancing and sweating and being present in one's body, but also with others. And so this idea of sort of sequencing an environmental sample of what connects us was what was the most important for me. So the project has taken a couple different heads. There's a few different elements to the project, but it's coming together. It's exposing hopefully next month at World Creation Studio, and then next year there's a few more bookings in town, which I'm excited about. So hopefully you can experience that, because eventually the project will be these sorts of live sequencing events will be translated into laser projection visuals and then emitted back onto a dance floor. So we can sort of relive these informatic experiences that connect us.
That is absolutely mind-blowing, and I would love to partake, because this is, again, you're grappling with these interactions and this like conversation that really we're all interacting, we're all having these sort of connections even at a microbial level, right? And taking that and then relaying that back into a visual scale. My last question is, what do you expect people to take from this project? And how does that relate to creating bridges between arts, society, and culture?
I mean, there's so many things I hope people can take away from the experience. I think for me, I remember the moment that I felt that connection point. It was so invigorating for me that I was obsessed with recreating it for someone to re-experience, but also being able to try to utilize what we know so much about our microbiome and all of these things that connect us, sometimes in very nefarious ways, but most importantly, I think it's pretty beautiful how it connects us all. And yeah, I think that there's, even though many people would think that a sweaty dance floor might be a sort of wasteland for microbial life and you're going to catch something bad, but I think some of the most beautiful healing experiences I've had in my life have taken place in those spaces. So I can only imagine what trying to recreate that experience for someone else could do, hopefully. Fingers crossed.
Definitely. Well, thank you so much.
Thank you.
Thanks for coming. We'll be moving into breakout discussions. So if each of our panel, interviewee, interviewer duos could take a table, we'll be having separate discussions. We'll give it around 15 minutes of discussions, if there could be a note taker, and then at the end, we'll all relay back and share what we've touched on. Does that sound good? Okay.
Thank you.
I'd just like to invite everyone back to the main seating area, I guess. We can wrap up hope everyone had some good conversations. So now we can just join back and kind of discuss what we've learned from all of these cool projects.
To help you
I can't hear
I'll
you.
give everyone a second to sit down, and then we can invite
Thank
whoever
you.
wants to say something
Okay,
about what
thank
they
you.
learned.
Thanks. Bye.
Yeah, do you want to jump there? Turn.
I'll give a little wrap up of what we talked about in the group back there. We talked a little bit about how art permeating into science as an artist can be a little tricky because of set ideas that scientists may have, and really how important it is to emphasize that there has to be some kind of fluidity. We talked about the ideal structure for these science-art collaborations, and we thought that the best play space for these types of conversations and interactions would be in a lab that is specifically designed to accommodate art practices. We also talked about a bit about different timescales, about how art moves at a different scale, at a different time scale as science generally does, and how it might be important to actually scale back and think about things in a wider time frame without as much urgency, without, you know, the perish or die type of mindset that might really lead to interesting and future approaches. That was kind of a summary of everything we talked about. Maybe someone wants to
Yeah, so with Emma, we talked a bit more about the barriers many African students face in getting access to synthetic biology education. Specifically, the equipment resources are hard to come by. It's really mostly Egypt in the north and South Africa in the south that have centers for synthetic biology. And so Emma was saying, like, in the future, it would be great to have educational centers in the west and east of Africa, so that students in countries all over the continent can have access. We also brought up that to go to the IGM competition, it is really expensive. And Emma actually reached out, I think, a couple times to the board of directors asking if there could be fee waivers from countries in Africa that don't have the funds to send their students there. And they were told that it would not be possible, which is obviously really frustrating. And maybe we can put more pressure somewhere to help with that. Oh, he also did say, though, that there is actually a virtual synthetic biology competition that I think he's looking to participate in as a way to give that access to students in Africa. And yeah, did you want to add? Yeah.
I just want to add, in the context of Emma, you know, the challenges we talk about, equity, diversity, and inclusion. And honestly, I didn't know when I started training graduate students that to get an international student visa, you needed the $10,000+ to showing your bank so that you could support yourself. And after 30 years, the government has raised that to $20-something thousand to just get a student visa. It is a refundable fee or something you need to show in the bank. It means that in terms of inclusion in attracting our students, and I could not bring Emma on that budget, you know, we are only getting international students who come from wealth, even from emerging and developing countries. They're the ones with the deep pockets, and that's a really big piece for me in terms of inclusion as a Canadian society to people around the world, to equitable educational opportunities. And I know that Emma has just started, for instance, a master's degree in Europe, and that he's had to crowdfund just his travel, and is now crowdfunding to try to get enough to pay his tuition in advance of receiving a scholarship, because if he doesn't pay that before the scholarship is paid out at a set date soon, he loses his opportunity to study. So understanding those barriers, I think, is really important when we're talking about inclusion. And I went to the COP15 meeting of the Convention on Biological Diversity, and a lot of nations were begging for more access and more resources to even get the technologies and the learning and the access to those technologies, I think it's very important to recognize what somebody like him is doing in the face of such incredible barriers to accessing education even, you know, for his master's degree, which is in question if he doesn't raise $3,000 for tuition or £3,000. And it's a very big struggle that I personally have noted. And that's why his efforts are just so incredible, because there's just such a desperate need to get technologies out to all nations in all parts of the world. And we take a lot for granted in our systems here when we're talking about our training and our trainees in synthetic biology and other emerging technologies. We've got it, and it's easy enough for you as a master's student or an undergrad to get access to that, but a lot of places do not have any access at all. So I commend him on his amazing work and wish him luck in that journey. Thanks for your time.
I guess we're 10 minutes early, but that concludes sort of our segment. Please stick around, if you will, for some light conversation with our remaining interviewees. We will be back for mitochondrial drama very shortly at 4 p.m. if you want to stick around for that. Thank you.
Okay, thank you, everyone. As Zoe mentioned, we will be back at 4 p.m. with our next installation of this SynBio Expressions conference here. So we're going to be closing up the live stream now, and just a quick reminder that this is already available on our YouTube channel if you'd like to revisit and share it. Join us again in one short hour. We'll see you soon.